All right, today I'm going to go over the nervous system 1, the first set of lecture with the nervous system. All right, the nervous system, this is going to include nervous tissue, the brain, and cranial nerves. It's also going to be broken down. We break it down in a couple different terms: central nervous system, which is the brain, the spinal cord, and the membranes that surround those two parts, which are called the meninges. Then we have what we call our peripheral nervous system, and this is every other nerve in the body other than the brain or spinal cord. A couple of the terms here. Nerve is not an individual nervous system. It's a bundle of nerves, mainly the axons. And we find these usually in the peripheral nervous system, because an individual nerve cell is called a neuron. So when you say nerve, it's actually a group of neurons. Then we have a ganglion. And a ganglion is a swelling. You find them near the spinal cord, and these are where the bodies of the neurons are located. And so for a nerve, you remember it's a bunch of them together, and so it'll be a little swelling, and that's the ganglion, because there's a bunch of the cell bodies at that one location. As you see here, the ganglion are just outside of the spinal cord. Because the axons, we haven't got to that part yet, but the axons of the neurons are much longer. So again, they're color coded here. These kind of pinkish ones, flesh colored ones, are the central nervous system going down the spinal cord. Notice it stops right about here at the bottom of the ribs, of the thoracic ribs. Well, I guess that is all the ribs. And then below that, it's called the equinocaudal. Then you have nerves, and everything outside of that, these yellow portions, are the nerves. So again, two systems, central and peripheral. Peripheral system gets broken down into what we call sensory and motor. Now, sensory, these are also known as afferent. This is anything that's coming in. Now, they don't break it down on this picture with a diagram with other pictures below it here, but if you look, it says the somatic and visceral. So that means that some of these, the input is coming from the muscles, and some of the input is coming from like your internal organs. That's the difference between somatic and visceral. We'll cover that a little bit more in a moment. So then our motor, these are known as the efferent. This is our actions that occur from the input from that we get from the sensory side. Again, see, they're broken down into the somatic, which are the voluntary ones, these are like our skeletal muscles, or the autonomic, which is known as the visceral, okay? And these are ones that we do not consciously control. They happen whether we are thinking about them or not. Now, the autonomic gets broken down again into the sympathetic and the parasympathetic. And these two are antagonists of each other, so they work opposite of each other. Sympathetic in general causes the body to be ready for activity, to doing a lot of movement around, doing a lot of physical activity, I guess I should say. While the parasympathetic is conserving, your body's relaxing, resting, and then other things that we don't normally think about occur at this point. So this is a good flow chart to kind of remember, to review, and we're going to cover a little bit more of it as we go. But this flow chart here is a pretty good one to review for quizzes, tests. All right, so these functional and divisions of the peripheral. Again, sensory, these are the afferent, these are the ones that are coming in. Gets broke down into, again, like I said, the somatic, which these are the ones coming from our skin, our muscles, and bones. These are what we think about. And then we have the visceral, heart, lung, stomach, bladder. We don't consciously think about this. Okay, and that's the difference between. So this is more kind of an external thing, and the visceral is more of an internal one. But I don't want you to completely distinguish them that way. It's more about the conscious control of it. So that was the input. Now the output, which is the motor. Again, we have the somatic, and this is going from our central nervous system out to those muscles. This is the reaction, the movement that we cause. These are ones, again, we think about. The visceral, this is the autonomic ones we don't think about. So these are going to our glands, our heart, because we don't tell our heart to stop beating or to beat faster. So again, these are ones we don't think about. And remember, the visceral or autonomic gets broken down into the sympathetic, and again, increased heart rate, increased breathing, decreasing digestion, because if think of this one as kind of the fight or flight, because if we're fighting or running away, we're not worried about digesting. We're just want to get as much oxygen and blood to our muscles to run or fight. The parasympathetic on the other side, these are the ones that slow us down, so they're going to lower our heart rate, lower our breathing. This one is going to increase the digestion, so we get those nutrients and everything going out to everybody. So again, one is for action, one is for conservation of our energy. Remember the differences between the two. Properties of neurons, similar to like muscles. Muscles had 4 characteristics. These are pretty similar to that. Again, they have to be excitable. In other words, they're going to respond to some kind of stimuli. They have to conduct the stimuli along their membrane. We call these action potentials. And then pretty much all of them will secrete something. And they usually, it's a chemical here known as a neurotransmitter. And we'll talk about a few of them. We'll talk about more when we get into the endocrine system. But right now, we'll just talk about a few of them here. If we classify neurons by their functions, we have to label them a little bit different. So again, we have our sensory and our motor. So our sensory, these are, again, bringing stuff in. Light for our eyes, heat, pressure, chemicals on our tongue. Nose also, for that matter. Again, that's bringing the information in. Motor neurons are going to be sending the information or a response back out. We have interneurons also. All right, so again, you have your sensory that brings the information in. Your interneurons decide what to do with this information, and then the motor neurons is the action determined by the interneurons. Okay, so those are the 3 classifications of their function. So they're showing you, drinking the water, decide what you want to do. Well, you pick the water up and you drink it. Now let's go to the individual neuron. This is an individual cell. We have got to get the anatomy of it. So we have the soma, or the cell body, and this is where the nucleus and all the organelles are located at. We have what we call dendrites. These are it does kind of look like branches of a tree or a bush. They're shorter. We call these cytoplasmic extensions. So they stick off of it, and it'll have lots of these. Then we have the axon, and this was a bunch of these axons is what makes a nerve. These are, these send a signal away from the soma to another area of the body. And then at the end of the axon, there's only one axon, by the way. The axon terminal is at the very end of it. It's going to be a little swelling at the end. Okay. The space between the axon terminal and the next cell is called the synapse. Sometimes this thing is called the axon terminal, axon button, terminal end plate, something like that. You'll see lots of different things for them. And again, see, it's the synapse is the space between the muscle cell, which we've already talked a little bit about, a gland cell, or another neuron, depending on where it's going to. So here's a picture of one. Again, we have the soma, which is the body, where everything's located. You have all these short extensions. These are called the dendrites. You have the axon, which is much longer than a dendrite, and there's only one axon going away from it. You have these little cells. In this case, they're called these blue ones are called Schwann cells, and we'll get to those in a little bit. They insulate the axon. You have little spaces in between each one of these cells, and that's called a myelin sheath gap or a node of Ranvier. Then here again at the very end of the axon, you'll have all these little branches. And then at the very end of these little terminal branches, you have the axon terminals or terminal end plate or terminal button. Again, depending on your textbook or what instructor you're having who calls them. So I try to give you lots of those terms. All right, so types of these things. So we can have neurons that have what we call multipolar, and this is what we typically think of as a neuron. So again, it's got one axon, but it's got lots of dendrites, and we'll show you pictures in a moment. You can have a bipolar one where it's got one axon and one dendrite, or you can have a unipolar one where the cell body kind of sits off to the side, and the impulses that come to the cell and then get transmitted away from it don't actually go to the body of the cell. So here's what we're looking at. Your typical one we think is the multipolar. You have all these dendrites that receive the input, and it kind of goes to the body of the cell, and it kind of decides if it's going to send it on or whatnot. I say decides, put that in quote. And then the axon sends the information away from this cell. So again, this is a multipolar. Unipolar is you have one dendrite, and then you have one axon, and it has to go past the cell body. Excuse me, that's a bipolar. Unipolar, the cell body, it doesn't have to. So the dendrites receive it, and then it's passed on to the axon without going through the cell body. But on these other two types, it does have to go through the cell body. Just be aware of these. For our purposes, that's all we need to know. If you decide to take more classes, get more in depth on it, they'll discuss this more. All right. Neurons are very specialized. So they need what we call babysitter cells, and these are known as neuroglial or glial cells. And we have far more of these than we do actual neurons. And the ratio is approximately 50 to 1. For every one neuron, you've got 50 other cells supporting or helping take care of that one cell. And so neurons are very specialized, and because of that, they need all of that help. So types of them. There's 6 types. The first 4 that are listed here occur in the central nervous system. The last 2 occur in the peripheral nervous system. So our first one is the astrocytes, shaped like stars. Somebody thought so when they named them. These give it a supportive framework. They kind of attach to the capillaries and these form a blood-brain barrier because blood doesn't actually touch the brain. Okay, blood stays in the circulatory system. It doesn't actually leave the circulatory system. So this is just helping form that. Then you have microglial cells. These are like the immune cells of the central nervous system. And again, they phagocytize, which means they eat, and put it in quotations, eat dead tissue, other organisms that might get in there. Anything that gets in there is not supposed to be there. So these are the, again, kind of like the immune cells of the central nervous system. You have epidermal cells. These are in the cavities. And these have cilia. The cilia is used to circulate the fluid. Because there's in the circulatory system, we have the heart that uses pressure to move blood around. In the central nervous system, there is no pressure. It's a zero pressure system. So the central cerebrospinal fluid would just sit there otherwise. So we have these cilia on these epidermal cells that kind of brush the kind of like paddles or whatever you want to say to move the cerebral spinal fluid along so that it circulates throughout the central nervous system, and it later gets absorbed back, and that's how you make more of it. You have oligodendrocytes. It says they're octopus-like. Yeah, they're hard to see under a microscope. But these surround the nerve fibers, so these are the axons that they surround. They form a myelin sheath, and myelin sheath is it's kind of a fatty, lipid-based substance that coats these fibers and it insulates them. Okay? Now, again, these are the first four. These all occur in the central nervous system. All right. The next two are what we call satellite cells. They're in the peripheral nervous system. We really don't know a lot about these things. Just know they are there. And then Schwann cells. Now, Schwann cells wrap around the nerve fibers. So the nerve fibers, again, are the axons. All right. Again, they're going to produce that myelin sheath. This is only in the peripheral system now, not the central nervous system. So these are going to act very similar to the oligodendrocytes. They are very supportive for their myelin, and they help protect nerve fibers. Right here, you can see at the very bottom here. They assist in the regeneration of nerve fibers, and often you hear that nerve fibers cannot regenerate. Central nervous system nerve fibers have much greater difficulty and are much less likely to regenerate. Peripheral ones, depend upon the amount of damage, they can regenerate up to a point. So here are some pictures of it. Here's those astrocytes, how there's the blood, the circulatory system. They're having these feet that grab onto them. Here's your microglial cell that'll go through, and the white blood cell of it. Your epidermal cells, there's the cilia that circulate the cerebral spinal fluid. Here's an oligodendrocyte, and it's wrapping around several fibers, but it's making that myelin sheath. So these first four are in the central nervous system. These last two, the satellite, this purplish one, then we don't know a lot about. And then all these Schwann cells that we saw earlier, notice that they're blue because they perform basically the same function as the oligodendrocyte. But notice that there's many of these on one. All right? There's more myelination going on in the peripheral nervous system than there is in the central nervous system. Now, what does myelin do? It's an insulation. If you know anything about electricity, you know you want your electrical wires insulated, and it helps them transmit the impulse. That's what this here does. So it increases the speed of the signal, all right? So ones that have a lot of myelin can travel at about 3 to 15 meters per second, all right? The fastest of them can go up to 120 meters per second, and 120 meters per second is a little less than 300 miles per hour. So that's pretty fast, considering we're only, if you're a tall person, you're only a couple of meters anyway. Now, the unmyelinated ones, the ones that do not have a bunch of the Schwann cells or the oligodendrocytes, it'll only travel from like half a meter to about 2 meters per second. A half a meter per second is about a mile per hour, and 2 meters per second is like 4 miles per hour. So relatively speaking, it's not anywhere near as fast. But if you're going, again, you're only about 2 meters long if you're a tall person. So it'll still happen pretty fast, okay? Now, a little word. All cells have a resting membrane potential. Neurons, on the other hand, are specialized in that they can change this resting membrane potential very fast. Now, so that means that they're excitable. Resting membrane potential means that it can change its charge really quickly, which is how they send an impulse. All right, so let's look at some ideas about this here. Again, opposite charges attract. We kind of know that, positive, negative. Energy is required to maintain these, and we're going to get to this, but the resting membrane potential of the cells is that the outside of the cell is positive and the inside of the cell is negative. So that's a potential. When these charges move from one side to the other, you get opposite charges separate. When it moves from one side to the other, that's how we get to use energy. So we keep these things separated, and that's why we have the potential for it. Now, how do these charges work? Because we're talking chemicals here, not electricity, but it's similar. We have different types of gated, these are proteins that are in the plasma membrane of the cells. And don't worry so much about the three types. Just letting you know there are three types. So we have chemical channel ones. So these are the ones that are going to open with a neurotransmitter. What neurotransmitter we talked about so far? Acetylcholine. We talked about when we talked about muscles. Then we have voltage ones. So these are going to respond to when we change the amount of electricity from the charge of it. And again, this will make sense here in a little bit. And then you have mechanical ones, and these are when something binds to it, it'll change shape. Again, we talked a little bit about this in the muscular system when calcium bonded to, no, it was on the troponin. When calcium is, when the impulse goes into the T tubules and changes shape, there's chemicals involved in it. We just didn't talk about them. So that's how it works. All right. So when these gated channels are open, the ions, because this is where we get our charge from, diffuse very quickly. All right. So this concentration gradient, just like osmosis or diffusion, it'll go from a high concentration to a low concentration. And this causes an electrical charge for us. Okay, so that's but now we kind of reviewed some of that. I hope it's clear, but if not, please email me. We can measure these potentials with a voltmeter. Now, our resting membrane potential is approximately -70 millivolts. That means it's -70 on the inside of the membrane compared to the outside of the membrane. And that's what they're telling you. The cytoplasmic side is negative. So it's to be polarized. It's like a battery. You have a positive side and a negative side. The outside is positive, the inside is negative. We have a battery that gives us potential for energy. So we're going to call these the intracellular fluid and the extracellular fluid. That's what the ICF and ECF stands for. So extra is going to be outside of the cell. So we plug a voltmeter into one, and we have voltmeters where we can do this. One of your labs talks about this. The inside compared to the outside is negative. So again, there's our battery that's occurring for us. All right, and it's compared the two of them to the inside and the outside, it is -70 millivolts. Now this is caused because the extracellular fluid, so the outside of the cell, has a lot of sodium ions, a lot more than the inside. And there are some negative charges on the outside too, with chlorine. Okay? Now the inside has a lot of potassium. Now notice that they're both positive, so there's other things in play that help maintain this resting membrane potential that's positive and negative. But these two ions, the sodium and potassium, are the main ones that are going to cause any changes for us. And so they're balanced out with negatively charged proteins. And there's a lot of them inside the cell. They don't leave the cell. So they're inside of the cell. And that even though there is a bunch of positive inside the cell, there's more negative. All right. So what we do, we have to depolarize our cells. And what this is going to move the inside of the cell towards becoming less negative, so make it more positive. All right. And then we have a term called hyperpolarization. This is where we move the inside of the cell farther away from zero, so it becomes more negative. All right. And this will, trust me, this will all make sense in a little bit. These action potentials are only transmitted in muscles and the axons of neurons. So there's a brief reversal so that it becomes positive. There's a total of 100 millivolts change. So if we go from -70 and we want to go positive 100 spots, that means we become positive 30. So it doesn't really, there's not a big change, but it does change. So this action potential doesn't degrade, so it's transmitted down the length of the axon. And some axons in humans are more than 1 meter long. And we call this a nerve impulse. I'm probably going to call it an action potential the whole time for us. I'll name it an impulse from one time to time, but yes. All right, now how does all this happen? So again, in a resting state, we have sodium on the outside, potassium on the inside, and these channels are closed. To depolarize, we have to open the sodium channels, okay? Now what happens is all sodium comes into the cell. So we're moving positive ions inside the cell. And remember, the inside is negative. So if we're moving positive ones inside, it becomes less negative. Once it reaches about negative 55 to negative 50 millivolts, that's when we've reached our threshold. And threshold means we can't stop it. It is going to continue to go. Just like in muscles, once you reach the threshold of stimulus, the muscle contraction is going to happen. Same concept here. The inside will jump up all the way up to a positive 30. So going from negative 70 to positive 30, that's that approximately 100 millivolt change. Again, all or nothing. Once the threshold is met, the action potential is going to complete. It's going to go all the way to that positive 30. It's not going to go to negative 40, negative 30, and stop and go back. Once you reach that threshold, it's going to, the sodiums are just going to rush in. This is the depolarization. So remember, it's polarized. We have positive on the outside and negative on the inside. The positives move in, so it depolarizes. We end up where there's not a positive and a negative. It become positive. All right, to fix this, we've got to do a repolarization. So we close the sodium channels. We stop the sodium from coming in. It's like, nope, you're done. And we open up the potassium channels. All right, so now remember, we had a bunch of potassium on the inside of the cell. Now that potassium leaves the cell. Okay, so we're going to repolarize it, make again a positive on the outside, negative on the inside, because we're moving these positive ions out. So that's the repolarization. Now it's a little bit, the sodium goes out really fast, okay? So what happens is it tends to go a little extra, and we call that hyperpolarization. Because remember, the resting membrane potential is about negative 70. Well, the hyperpolar, because the potassium will go out really fast and it kind of stays open a little bit longer, we end up at about a negative 80, maybe negative 85 on that extreme end. So this causes a hyperpolarization, because hyper means more, so it becomes even more polarized. But it balances itself out in the long run. So here's a Dynaflow chart. You see up here, we have our resting membrane potential. It's flat. The sodium channel opens up. All right, here you see the sodium coming in. They're showing you the sodium ions going in. So that's our depolarization. Once it reaches that that positive 30, it'll close, and the potassium channels open up, and this causes us to repolarize. Brings it back down to that negative 70. But because it is going out pretty fast and it kind of hangs it open a little bit longer, we have that hyperpolarization. So if you look, this purple line right here is a little bit lower than that one, because again, it becomes a little bit more polarized, but everything will reset itself. So there's your charts. There's your resting membrane potential, your depolarization, your repolarization, and your hyperpolarization. Notice it does come back up. So after all of this is done, we have the charges correct, but the ions are in the wrong place. Because remember, the sodium moved in, potassium moved out. So that gives us our charges, but we can't go to another action potential until we put the sodium back on the outside and the potassium back on the inside. So after we repolarize, we have what these items called sodium potassium pumps, and there's lots of these in axons. They will pump the sodium back out and bring the potassium back inside, and that'll brings us everything back down to normal, to that negative 70 millivolts. But it puts the sodium on the outside and the potassium back on the inside, so we can have another impulse. They're showing you how this works over here. The sodium rushes in and it causes it to become positive on the inside. And so that through diffusion, it's going to slowly leak down the membrane. And it goes to the next spot, and so it turns it to positive, and it's going to bleed over, and eventually this one's going to be positive. Notice that it's becoming more negative outside because all those positive ions came in. And so the yellow here is showing you the resting membrane potential. So again, now once you reach it, so there's your depolarization, where it becomes depolarized, and we have that positive on the inside. I want you to pay attention to this gray area, though, as we go. So we're going along. You have your repolarization, your hyperpolarization. Now, this is sodium on the inside is this gray, potassium on the outside. We need those sodium-potassium pumps to flip that around and make it back to this yellow as we go. But because we have the sodium inside here, it's not going to, the action potential can only go one direction. It can't go backwards because the ions are in the wrong spots. Again, this is kind of a complicated process. So take your time, review it, read through your textbook a little bit, and help explain it. If you have questions, please email me. So now we've had our action potential. It's gone down to the end. At the axon terminal, the terminal end plate, terminal button, whatever you want to call it, it releases some neurotransmitters. We also have some receptors involved in this. So acetylcholine is one we talked about. All right. This one is secreted by both our sympathetic and parasympathetic, because acetylcholine is really with muscles, and we have our skeletal muscles, which we would think of with our sympathetic for our fight or flight, and with our heart too, for that matter. But we have our smooth muscles that are in our glands and our intestines and things that will be affected with the parasympathetic. We have a cholinergic fiber, and it's any nerve fiber that secretes acetylcholine. That's all you need for that one. Now we have these receptors. There's a couple of different types: a muscarinic and a nicotinic. These are usually excitatory, or they can be inhibitory. There's mushroom toxins that can affect these. Again, cardiac muscle, smooth muscle. Just note there are a couple of different types of cholinergic receptors, and that is a muscarinic and a nicotinic. And these are both part of this. So it'd be an M-acetylcholine receptor or an N-acetylcholine receptor. And that's good enough for these. Some more neurotransmitters. Norepinephrine. This is almost completely in the sympathetic, again, that fight or flight aspect of it. So you have your adrenergic fibers that secrete the norepinephrine, and then you have your receptors that bind the norepinephrine. There's a couple of types of these. You have an alpha androgenic and a beta androgenic. Again, one's usually excitatory and the other's inhibitory. So let's jump into the brain real quick. This will be a short little portion of it. Looking at some of the features of the brain. Rostral is going to be leaning towards the forehead. Nose is what it really means, but it's towards the forehead. Caudal's more towards the back, so the tail. And the brain is made up of three main parts: the cerebrum, the cerebellum, and the brainstem. The cerebrum is broken down into different lobes, so frontal, parietal, temporal, occipital. Cerebellum's not just one thing, and brainstem's got a couple parts too, and we'll get to them. So on all of this, this is the part I'd really like you to know. All of these with the squiggly lines and all the wrinkly portion, this is all the cerebrum. And it's broken into, again, your frontal lobe, parietal lobe, occipital lobe, and your temporal lobe. Then this cauliflower looking thing, maybe, I'm not sure, is your cerebellum. And then you have the brainstem coming off of it. The brainstem has a pons and a medulla. The medulla really controls the breathing, your heart rate, your body temperature, things like that. Cerebellum is really about your fine motor skill to be able to type, play a musical instrument, tie your shoes, gives you fine motor coordination. Gross motor coordination is done up here with the cerebrum. The frontal lobe is the last portion of the brain to develop, and this is your higher thinking is what this is, what we call our executive area. This really doesn't fully develop until you're approximately 25. This gives you more logical thinking. Now, those of you who are teenagers or early 20s, you're probably going to do more of your thinking with the temporal, which is part of the limbic, and where it does feelings. I'm not making fun of anybody on this, but that's why teenagers are more emotional and react emotionally, because they use their limbic system more so. Their actions are dictated more by their feelings than with logic and a higher order of thinking, which occurs in the frontal lobe. But again, that's not developed until about 25. All right. We have our parietal lobe up here. This little region back here is called the Wernicke's region, Wernicke's area. This one helps you understand. No, this one helps you speak. So if you have kind of if your damage is here, then you might want to say hello, but might say cauliflower instead. The words you produce aren't what you're thinking. And over on this area, and this is what we call the Broca's area, and this lets you understand speech. So these two areas work together with speech. So that's, again, parietal is that language. Just want to throw that out there for you. All right, so brain development. We have these 5 regions here. All right, and now we're not going to go into a lot of detail because it's beyond the scope of this course. The telencephalon here, this is going to form the cerebrum, the main portion of the brain. The diencephalon, this is going to form kind of the inside where the thalamus is, which is kind of directs messages along. The mesencephalon, this is the kind of the midbrain, which is near the thalamus. The medencephalon, this will be the pons. The myelencephalon, this will end up being the medulla oblongata. And so yeah, here, see the telencephalon develops into the frontal lobe. It's the last one to develop, all right, which is, again, why younger people tend to react more emotionally than elderly people, because their frontal lobe hasn't developed completely yet. A few more things. The rises of these folds, the crests are called gyrus, and the grooves are called sulcus. And we have main, so I'm gonna back up some for us. So like right here, this is what we call the central sulcus, divides the frontal lobe from the parietal lobe. There's another, a big sulcus right through here that would divide the parietal and the temporal. There's another one back here that's going to divide the occipital. And they're kind of vague, to be honest, hard to see them. The biggest one there is that central sulcus, though. Well, you also have this longitudinal fissure. It's kind of like all these. See the crests, that's the gyrus, and the grooves are the sulci. Down here is, again, that cerebellum, which is a coordination. And they're showing you the gyrus on top, sulcus is on the inside. Here's the internal view of the brain. If you look at, so there's that, there's your thalamus, here's your hypothalamus. This kind of, the hypothalamus communicates a lot with the pituitary gland. Technically, the pituitary gland is not part of the brain, even though it is right there really close to it. It's more of the endocrine system because it controls hormones. And the hypothalamus communicates to the pituitary on that. So here's your pons. This kind of helps direct things, impulses going to the brain or away. The thalamus does that too. The medulla, again, this one has some vital functions in there it'll tell. Let's just focus on the heart for that. It'll tell the heart to speed up if it needs to beat faster, or to slow down if it needs to beat slower. The heart will beat all by itself. It doesn't need anything else. But this tells it to, again, breathe more forcefully, less pressure. It also regulates our breathing. So it tells us to breathe more if we need more oxygen, or it will breathe more if we need to get rid of carbon dioxide. Slow down if we're getting rid of too much of it, because carbon dioxide is really what dictates it. This and the cerebellum, see this white stuff through here that kind of looks like a bush or a tree. That's called the arbor vitae. It's got some vital functions in it. But main thing I want you to know about the cerebellum is that this one coordinates muscle function. Again, for typing, playing an instrument, flute, saxophone, whatever, piano for that matter, and everything. Gives you the fine motor skill. We have the meninges. So these are the membranes of the brain, of the central nervous system. There was a thick one that goes right up next to the skull. This is called the dura mater, which translates to tough mother. Then there's a thinner one below that called the arachnoid mater. And a real thin one that is basically in contact with the brain. You see it coming down. This is called the pia mater, which means delicate mother. And somebody named this one the arachnoid mater because you see these fibers through here, makes it kind of look like a spider web, I guess. And that's why they're named here. You have a space between the dura mater and the arachnoid mater called the sub, which means below, dura. So subdural, if you ever watch a medical show, you hear subdural hematoma, that's okay. It's a bruise or a blood clot that's below the dura but above the pia, or it should be above the arachnoid. Then you have the subarachnoid, which would be between the arachnoid and the pia. So just keep those in mind. And these are continuous throughout the brain and the spinal cord. Here's some more. You see the subdural here. Now they're talking about ventricles. This one here is really showing that. And the ventricles are where the cerebral spinal fluid is produced, and it flows. They're showing you the flowing of the fluid coming down, and there it goes through the central canal, through the spine, around it. And so it keeps the central nervous system bathed in this fluid, which helps it because it's really soft. And if it was, it would actually collapse under its own weight if it wasn't floating in this fluid. But that gives us oxygen and nutrients, takes waste away from it. Here's part of the brainstem. You see this the diencephalon here with the thalamus and hypothalamus. This will be up into the midbrain, and it comes down, and you have your pons and medulla down lower. All right, so there's the pons and medulla down here lower. Then it becomes the spinal cord. Again, there's that cerebellum with the arbor vitae. Again, main thing I really want you to know is that this is for muscle coordination. These pictures show you the larger it is, they're showing you what part it's going to. This is right along the central sulcus. The red is going from the front to the back. So the red would be in front of the central sulcus, and the blue would be behind the central sulcus. And they're showing you on so this is the higher order area, the cerebral cortex, which is the outer area of the cerebrum, or the cerebrum. The larger the picture of whatever it is, the more it controls that. So that's why you see things in various sizes. All right. So this again over here, this portion of the brain is going to control a lot of the facial movements. This has got a lot of hand movements here, a little bit of feet on the left side, a little bit more controls it on the right side, so forth. All right. Here's that Wernicke's area I was telling you about. This is associated with making speech, language, and over in this area is where the Broca's area is, and this one here is understanding language. So if you have damaged here, someone could be speaking to you and it would be a foreign language. You would not be able to understand what they're saying. Even if they're speaking the same language that you normally speak, you wouldn't be able to understand if there's damage here. So again, these two areas work together. That is it for Nervous System 1 lecture. Again, questions, email me.